So today we're going to talk about SLIM, if we get to it, overlap extension, and Gibson cloning. So let's go right in. So yesterday we talked about restriction enzyme cloning. And I showed you guys how you do it. I talked about the protocol, talked about the, this is usually, restriction enzyme cloning is usually the first thing you try. It's like the classical way to clone. But there's a lot of problems with restriction enzyme cloning. What are some of the things you guys can think of? Yeah, you have to match your map. So if you want to clone X into Y, you got to match the maps, and the restriction enzyme sites have to match. And that means X cannot have those sites, A or B, in its sequence. Otherwise, it's not going to work. So restriction enzyme cloning is sequence dependent. You're dependent on the sequences that you are using, the order of the base pairs. There's also all kinds of other limitations with restriction enzyme cloning. So that's the main one. The second thing is restriction enzymes are expensive. And if you start working with kind of rare restriction enzymes, it can get really expensive. And you got to buy these repertoires of all these restriction enzymes from these companies. So it can be expensive. It's another limitation. It leaves scars. What's a scar? Yeah, where the sticky end has to be. So if this is gene X and you put C, the restriction enzyme site there, you're literally adding in a little sequence there. So if it's Jo1, it's CTCGAG. You can't get rid of that sequence. It's there. That's a scar. And imagine if you, sometimes you need to do restriction enzyme cloning of two genes fused together. So if you need to fuse X and Y together with restriction enzyme site, now you've actually added in two codons, CTC, GAG. I don't know what those code for, but you've actually added into that. If this is a fusion, if here's the promoter, here's the ATG, here's the stop, and you fuse X to Y, you've actually added in two amino acids. That can alter your protein. You might not want that. So these are what are called scars. They're scars that are dependent on the cloning process, and oftentimes you can't get rid of these. It's a product of the way that you build the construct. Those are the limitations of restriction enzyme cloning. So people have developed ways to overcome these limitations, because there are cases where you can't mess with the sequence, or you just stuck with the sequence that you have and restriction enzymes don't work. So people have developed ways to do cloning, and also not only cloning, but manipulations of the DNA that are independent of restriction enzymes. Okay, so we're going to talk about one of those methods in detail, which is called SLIM. And then at the end, if we have time, we'll talk about more methods that are very similar to SLIM that are used for cloning, like Gibson cloning and overlap extension, PCR. And I would encourage you to watch the movies that I attach. I think, yeah, I have a watching assignment today. There's just 5 minutes of movies. Watch that on YouTube. What?
Are the scars basically like time that don't go away?
Yeah, so let me show you the mechanism of how they get added in. So let's say you want to clone X, you design your primer, and we talked about how you need to add that restriction enzyme site. So we'll do BAMH1 GGATCC, and on this one we'll do Jo1 CTCGAG. And then in your plasmid, you also have GGATCC, CTCGAG. Okay. So when you do the restriction enzyme cloning, you cut this, and it will cut off, I think it's, some of these restrictions are different. I don't remember exactly where they cut. But this one, in theory, would cut like here, And so you'd have a sticky piece where on the other, let's say the other strand, you would have C, C, T, A, G, G. This is all matching this. Here you would have G, A matching this, G, C, T, C. Okay? And what happens when you cut, you produce, now this is a sticky end. These two base pairs and these two base pairs are looking for matching sites. I might have drawn this a little bit incorrect with 5, 3, 3, 5, but you get the point. And the same thing happens down here is the sticky end here would be the GG, so it would be, right? And so what you're doing is you're producing this, which matches this, and they can find each other. Same thing happens down here. And then, so then when that gets ligated in, you're now stuck with GG, AT, CC, gene X, CT, C, G, AG. So these are the scars. We're stuck with that in the sequence. It's there. Sometimes we don't want that there. That could mess up your protein, or sometimes it's just sometimes it's bad to have that. So those are what you would call scars. Okay, so let's talk about SLIM. So in brief, SLIM is a way to add in a tiny sequence, or to delete. You can delete big sequences, delete sequences, or to mutate sequences. Okay, and we're going to talk about that. And by understanding SLIM, you'll understand all these other PCR-based, these are PCR-based assembly methods. Okay, so let's talk about SLIM. SLIM is hopefully you guys read the paper. It was a very short paper, right to the point. Site-directed, ligase-independent mutagenesis. So let's go through these. What's site-directed? We talked about that.
It's based on sequence in the DNA.
So you're choosing a spot where you can do these things. Ligase-independent, what's that mean? No ligase, does not need ligase. Yay, you don't have to buy ligase. It's good. And then mutagenesis is changing stuff. So like deleting something, adding something, and mutating a sequence, okay? So this is a PCR-based method that is site-directed, ligase-independent, and allows you to do mutagenesis, okay? Let's see. So here's a question. How is it ligase-independent? What does that actually mean mechanistically? So normally we talked about what's the role of ligase. If you clone something in, steals the phosphate backbone. So when you just anneal with the sticky ends, the phosphates are not sealed, okay? But the ligase, the T4 ligase that we use, the enzyme, is the enzyme that actually seals the backbone. So the way that this is independent is most of the time when we talk about the restriction enzyme protocol, there's actually a step, which is what we would call in vivo, no, not in vivo, in vitro. What's in vitro mean? In the tube. We actually do a ligation step in the tube where we add, remember, the reagents we had the ligase, we had the buffer had ATP, we add our template, we add our insert, the backbone, blah, 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 blah. We all add all that into the tube and we do this in vitro ligation step, okay? Well, in SLIM site-directed ligase-independent mutagenesis, what we do is we have a bacteria, and what we do is we transform in a circularized plasmid that's annealed together. We transform that in. It's not sealed yet, but we transform that in, and then the bacteria seals it itself with its own enzymes. That's how it's actually ligase independent. Okay. Why would you use it? Why? So like, what do we want to do when we usually do this? Okay, oftentimes the first thing we do, like I said, the first thing you do is usually a restriction enzyme clone something. So let's say we got gene X. We cloned it in, okay? But then we realize, well, this isn't really that helpful. Maybe we want to insert our his tag, his 6. And I told you last time, you can actually add these tags onto the primers of when you restriction enzyme clone is 6. You can actually add that into the primer. So you might ask, well, why wouldn't I have just done that before? Well, maybe you did do that before, and maybe you found out that the his 6 tag was actually a really bad tag. It messed up the function. So now you want to switch it out. You don't want to do a whole new cloning. You just want to switch it out. Maybe you want to switch it out with a V5 tag or a flag tag. Okay, these are just other tags that you can use. So maybe you want to switch something out. Or another common reason is maybe you're studying, so you're studying gene X, and you think this region right here, the C-terminus refers to a protein, but it's coded for in the DNA. So maybe you think the 3' end of the gene codes for a really important part of the protein, then you want to do a negative control. So the best way you can do a negative control is by deletion analysis. So you can actually delete that sequence and see how that changes the functionality of the protein. So maybe you want to make a new clone that's just gene X with this region deleted, okay? So SLIM is a way for you to do that. It's a way for you to get rid of, erase the whole thing. It's a way for you to get rid of that sequence that was right there.
When you have like knockout animals that have a gene that's been deleted for the same reason, is this the same method?
This is upstream in the pipeline. If you're talking about like deletion in a mouse or deletion in yeast, that would be the best analogy. So we talked about how you would delete a gene in yeast where you would amplify a cassette, something that you can select for, and you would put homology regions on the 5' and the 3' end that match the gene that you want to delete. And then you can select for a recombination event, which then inserts that cassette into that genome. So that's like a chromosomal modification of an organism. This is a plasmid modification. Sometimes, so here's why I'm saying this is upstream of that. Because sometimes to build this cassette, this needs to be built, right? You need to build a plasmid that encodes those homology regions and the cassette. And sometimes you do that with restriction enzyme cloning. You would have to clone in the cassette. You would have to clone in these homology arms. Sometimes that is then, this plasmid might then be modified by SLIM. Does that make sense? So it's kind of upstream in the pipeline. A lot of times we want to make manipulations to plasmids before we're ready to do like a chromosomal deletion or a chromosomal insertion. So SLIM is a way to delete on a plasmid, not on a chromosome of an organism. Those are different. Not on a chromosome of organism. Does that make sense? Different methodologies. But it might be upstream in the pipeline of you building a plasmid that might be used for deletion in an organism. I guess it is a deletion of an organism because it's in a bacteria, but you get the caveat here. Things get nuanced. So oftentimes you want to insert an epitope tag. Sometimes you want to change a sequence. Sometimes you want to delete a small segment. A lot of times you do this to build a negative control. So like I said, if you're doing in vitro biochemistry, you're studying protein X. It's an enzyme. You're studying that enzyme activity. A lot of times you'll have your positive, which is your wild type, and you measure the activity by some experimental methodology. And then oftentimes you'll have a negative control where you have broken the enzyme, and now your activity should be equal to 0, or at least should be the activity of wild type should be greater than the activity of your negative control, right? So oftentimes this is important for just designing ways that you can build negative controls. Again, I've said this before, 90% of like your work in molecular biology is building reagents. So you're going to spend, you don't just have a negative control all the time. You actually have to build your negative control. So this is a protocol by which you can do that. Okay, so we're going to walk through, I'm just going to briefly walk through what happens, and then I'm going to walk through actually like how you would design the primers and actually like lay it out for you. So basically what happens is you have a plasmid, okay, and you design 4 primers, okay? There's 2 forwards. In the paper, it calls them something like this. And then there's 2 reverse primers, okay? And the catch is, so if these are your reverse primers, if these are your forward primers, actually, hang on. Wait, did I do this wrong? Shit, I did this wrong. Hang on. So these would be your forward. You have your forward, the shorts, the reverse short, and the reverse long, or reverse T, whatever they call it. There's a long one and a short one for each pair. And the trick is that these primers have these extended regions that match each other. So you have 4 primers, 2 forwards, 2 reverses, and there's 2 short primers, a short forward and a short reverse, and then there's 2 long ones, a long forward and a long reverse. And the key is that the long primers have this little region that matches, okay? And that is what creates sticky ends that can anneal. So you're essentially mimicking what a restriction enzyme does, but you're just doing it by building it into your primers. And then what happens is you actually PCR all the way around the plasmid. Okay? And let's look at this figure from the paper. I put this on your thing that you should really study. You should really study this figure. Because if you understand this figure, you understand SLIM. Okay? So let's look at this figure. So this is what I just drew. There is 4 primers, a forward that has a long matching overhang, a reverse that has a long matching overhang, and then there's a short one and a short forward, okay? And if you think about, if you just do like I think this is like basic probability, if you look at all the possible combinations of PCR amplifications that can be made, so you PCR all the way around the plasmid, and in each case, one strand is only primed by 1 primer, okay? Now you look at, what you're gonna have is a mixture. You're gonna produce a mixture of products. And if you look in the paper, they tell you all the possible products that you can form. So you can form one product that looks like this, that has these overlapping things right here on one side. You can produce another product that has overlapping things on one side. So this would be if one of these primers ended up with one of the short primers. This one would be the reciprocal of that. You can also have a product that would amplify if it was primed by both the ones with overhangs. So there's both overhangs, okay? And you can also have the double short. The double short, where it's primed by 2 double short primers. So these are all the products, the mixtures. You'll produce a chimeric mixture of amplicons in your tube, okay? Then the secret is, what happens is, when these products, what you do is you denature them. You denature them, so that means you heat them up for a second and then you cool it down. They're gonna shuffle, okay? So when you heat them, you'll break them all apart. You cool it down, they shuffle, they mix back together. And then what happens is you will produce hybrid products that look like this. So this is a hybrid product where one of your pieces came from here. I'm drawing so much red, but you see that, right? This one came from here, and this one came from here. So they shuffle. And now if you look closely, this one has a sticky end, and this one has a sticky end, and those sticky ends can match. So now what happens is the plasmid circularizes circularizes, because those sticky ends, it just binds to itself. Does that make sense? So you produce this plasmid by PCRing all the way around the plasmid, and the sticky ends match, and so it can circularize, okay? So this is how you would do an insertion. So in this case, they're showing you an insertion, and what gets inserted is whatever you make up to be the matching overhang. That actually will get inserted right there into your plasmid, okay? So let's think about a deletion. A deletion is where instead of inserting something new, you make the sticky end match a part of the existing backbone. And then you match that to this one, okay? And as long as you're leaving out this sequence right here, when that thing recircularizes, it's going to recircularize with the deletion right there. And it's going to match there, come together. Does that kind of make sense? So look at this paper and study this figure. This is going to help you understand the mechanism of how this is happening. And it's basically, if you just understand that you're PCRing around the plasmid, that's going to help you understand. And the key is you're producing overhangs by building them into your primer. And I'll show you how to build them into your primer, and then you'll really understand what I'm talking about. Let me just make sure. Okay. I'm going to pull up my Word document, and now I'm going to really show you in detail how this happens. Hopefully I don't cancel the crutch. Okay. Pull up that Word file. What's going on here? There we go. Pull up this Word file. Okay, so let me walk you through how you actually would design primers for something like this. Okay. Step one, decide what your goal is, okay? So I'm going to give you an example of, I want to insert a C-terminal flag tag onto gene X, okay? So this is where I wanted to do a Western blot, and I couldn't blot for my protein because I didn't have an antibody. So I just want to insert a fused epitope tag so I can do a Western blot on my protein, okay? So that's what I did. So let me show you how I did that. And I'll walk you through some of these notes. So on some of these tags, if you're inserting a tag, you have to be very careful. So this is a his tag, right? This is a sequence of a his tag. Now, what's one of the problems if you were to make this one of your overhangs? Cat, 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 cat. The problem is the same sequence could shift over, right? Because it's a repeat sequence. So you could have something shift over because it still matches as it shifts. Does that make sense? So you have to be very, very careful when you're inserting something that's a repeat. You want to be careful to change up some of the sequence. So this is the same tag. It's the coding for the his tag. But here I've made cat, 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 cat. So I'm using alternate codons for histidine to make sure that that repeat sequence can't shift over. Does that make sense? Okay, so that's just a note. Be careful if you're putting in things that are repeat sequences. Okay, so the first thing that I did is you map out the sequence, right? So I knew my gene, or you can get it from NCBI. You get the sequence, okay? And here I've got 3' upstream of my stop codon. So here's my stop codon, tag. Note the stop codon. That's the stop codon of my protein that I'm studying. And here's the sense strand, 5 to 3. Here's the, Here's the antisense strand, 5 to 3. And I've mapped that out, okay? So if you, that does not match. This is my error. What?
You reverse the second line.
If I reverse this. So what I did, so here's what I did. Here's my problem is I didn't do the right function. Let's do this. Let's just do that quick. That'll be a good exercise. So here I caught an error. You've seen my logic. Let's go to reverse complement. Reverse complement. So you're saying all I need to do is reverse that sequence. So I can change this to reverse. And then let's test this. Good catch. Fix that. Okay. Now, so what I did is I mapped out the 5' to 3' sense strand, and now the reverse strand, which was fixed. It was an error, okay? And this is my stop codon. I want to insert a flag tag right here, okay? So you get the basic idea. That's what I want to do, okay? So let me show you how to design the primer. So Slim requires the 4 primers, 2 shorts, 2 longs, and I'll show you how to design them. But first, look at the target, okay? I'll fix that later. Here's the flag tag. So this is what I actually want to insert. So this is the amino acid sequence, DYK, DDVDK, okay? But I need the nucleotide sequence, okay? And I can actually choose this, right? Because there's multiple codons for Ds, there's multiple codons for Ys, multiple codons for lysines, okay? So how would I choose what the sequence is going to be? How would I choose that? Does that make sense? Like, if there's 4 codons for a particular, I don't know if there are 4 for D, but let's say there were. Let's say there's 4 codons for D. How would I know what to make my sequence? Does it matter? Yeah. You're going to want to check the organism. So I don't want to put something that's a bunch of bad codons. So what I would do is I would go to Google. I would search E. coli. This is going to be, this was actually in yeast. So I would search for a yeast codon preference table. I don't know if there's, I don't know if there's going to give me this. Let's see. Let's search images. Here's E. coli and yeast. I don't know how to interpret this. Let me give you the E. coli one, because the E. coli one is very easy to read. This is what I designed my art piece from. So look, I would go, if I would pick, I'd look at this codon table and I would say the Ks. Which of these Ks is more favored? The AAA, 3.8%. So of all codons in E. coli, 3.8% of those are AAA. And the other one that codes for lysine, AAG, is basically half that. So it doesn't like the AAG, it much prefers AAA. So let me see if I picked that. I did. AAA. But look, that's the K. But then I picked AAG. I split it up. I split the difference. I don't know why. But that's how you would choose the sequence. You could choose, in theory, anything you wanted. But in general, you would go to look at a codon preference table. That's how you would know what sequence. So this is the sequence that I need to insert into the plasmid. Thus, this is going to be the sequence that hits those, that is on those overhangs. Okay, that's going to be the floppy sequence in the overhangs. Okay, so now the other thing is the sequence, the tag must be in frame. What does that mean? Yeah, you got to go by 3s. If you produce a frame shift, your protein might never end. It might just keep translating. So when I say the tag that you're inserting needs to be in frame with the stop codon, that means whatever I paste in has to be, the length of it has to be a multiple of 3, okay? Otherwise, you are frame shifting. So here's the coding region of the gene. And here what I did is I literally just took the sequence that I got from here, I copied it, and I pasted it right before the stop codon. And then I just checked 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. It matches the frame. It's in frame. And then what I'll usually do is, once I have the sequence laid out, I would copy the whole sequence, go to ExPASy translate. Have I shown you that yet? Okay, let me show you that. So just in case I made an error, what I would do is I would go to, this is another important website, ExPASy translate. Okay, I would paste my sequence. I'm going to change this to compact. So this is just going to translate my sequence into amino acids. Translate sequence. Now there's So here's frame 1. So now look, I'm checking to make sure I didn't make any errors. Sometimes if you type something out, you'll make an error. And here's what I'm checking is, here's the end of my protein. I know this by heart, RGSR. That's the end of my protein. And I then inserted my tag. Look, there's my tag, DYKDDDDK. And then the dash is a stop codon. So I verified that it's in frame. I just did that visually by having the computer program translate the sequence from the base pairs to the amino acids. So you'll use this website a lot, ExPASy Translate. If you need to find a codon, we'll pull this up again later in a subsequent lecture where we want to mutate a very specific amino acid. You have to find the codon for that amino acid. And so you use ExPASy Translate to pull that up. Okay, so here's what I want to make. Okay, this is what I want my sequence to become. We all understand that, right? We get the purpose. Okay. Let's see here. So now I'm showing you how to design the primers. Okay. So the first thing is the long primer needs to have that region. Let's pull up that paper again. Let's study this paper. The forward long primer is basically, if you look at right here, it's the sequence that matches the plasmid, and then it's an overhang that does not, right? You see that, right? The dashed line, that's the overhang that does not. So let's go back to the Word file and let's actually do that, okay? So what we do is, here's the sequence that matches the plasmid. So this is the stop codon on my gene. I haven't changed anything. In this green, yellow sequence, nothing's been changed. The green is plasmid backbone upstream of my stop codon, okay? And the yellow one is my stop codon. So what I'm gonna do is I would take that, I would copy that sequence, and I got that from here. So what I did is I started here, and then I obeyed the CG clamp rule. I wanted to end with the G or C, right? Same thing in primer design. So copy that, and let's test this. Let's look at that primer. How do we test our primers? What's the website? OligoCalc. Oligonucleotides. OligoCalc. So let's check this out. Let's see let's check to see what it looks like. So here's the part that's going to anneal to the plasmid. The other part is just a floppy thing that's not going to anneal. So what I'm looking for is a temperature. It matches. So remember, we want our primers between 55 to 60 degrees Celsius, salt adjusted. So 57.6 is pretty good. I would check self-complementarity. Good, no hairpins. But even if there isn't much I could do because I'm restricted. I need to insert this tag right before the stop codon. So I don't really have many options. But I would check this, okay? So the annealing temperature looks pretty good, okay? So this is the part that's gonna be annealing, and I'm adding this on. So my forward primer is literally that. Does that make sense? That's our first primer. We've designed our first primer. Now the short primer, the short forward, is very, very easy. The short forward just matches the part from here to here that anneals. So I just copy this and paste it down here. That's my short primer. Okay? And if you look at the paper, again. The short primer is the exact same sequence as the annealing part of the forward long primer, right? Okay, so we did that. Okay, now let's do our reverse primers. Okay, we know that our reverse primer, look at the paper, the reverse primer is now basically literally at the point where we're inserting but going to the left, okay? And the overhang has to be the exact same sequence as our forward long. Okay? So here's how you make it. So if you look up at the top where you want to insert GGAT going this way. GGAT going this way. Okay? So I first found something that could anneal. And what I would do is I would take this, try to follow the CG clamp rules, and I would copy that, and I would see what the annealing temperature of that was. 62. It's a little high, but it's okay. It's okay to be a little bit high. So that's fine. no primer, no hairpins. Okay, so then what I did is I literally just took that sequence, the green sequence, and then I pasted on the tag. This is the part that's going to match. But here's a question. Am I done yet? Is this my actual reverse primer? You got it. Yeah, you got to take the reverse complement. So then what I would do is I would take this, copy. Shoot, I got rid of it. Go to that reverse complement website. Paste it in. Now here, I want reverse complement. Now this is actually going to be my primer, so let's test it. Let's look and see if I did that. Double check my work. Does this match this? Yes, it does. See? So I reverse complemented it. And now what you're looking at, you're still looking at the same thing, but it's just reverse complemented. So now the part that anneals is on this side, which I marked in green. And this is actually the part that's going to anneal. This is your flag tag, but reverse complemented. So this part is going to bind exactly to your forward long. This should bind exactly that. Yeah, see, look, CTT matches CAA. You see that? G matches here, A matches here. So this matches, okay? Now reverse short, very, very easy. Same process. Just take that part that anneals to the backbone, paste it below. And if you look at the figure from the paper, look at the reverse. It's the exact same thing as that reverse part that anneals to your backbone, okay? So we've just designed primers for a slim insertion, okay? So then what I would do is when we need to order the primers, I would paste them like this. These are all the primers that I designed. I would now check these. So I've made long primers that have added on sequence. So now I'm going to recheck these things that I added stuff onto for hairpins. Check for the hairpin, and I guarantee you this one is going to, remember we ignore this because it's not actually the part that's annealing. It's actually something longer. So ignore that 77. And there's hairpins, okay? So there's two things we can do, and I talked about this. One thing is, let's look at where that hairpin is occurring. One of those hairpins is occurring in our flag tag. So if it was possible, I could choose a different codon if it would get rid of that hairpin. Does that make sense? So I could do that. Or I could just say, I'm going to notate that that this primer is going to work better if I do hot start. That's why I said usually just always do hot start. And we talked about hot start PCR last time. So hot start's going to get rid of that hairpin. So I just notated that. Let's check this other one. Let's just see what happens for a repetition. So I go to here to oligocalc, calculate. No hairpins. I don't really care about this stuff. No hairpins. So that one's good. So here I notated. Oh, there's a hairpin. Okay. Now we have a little bit of time to talk about another important part of this protocol. Okay. So what goes into the reaction? This is just standard PCR. Okay, you have your template, your backbone. That's what you're going to insert into. Okay. You have your 4 primers. So this is the reagents that go into this tube. You have your buffer for your fusion polymerase. You have your DNTPs to load the polymerase, some water to make it to whatever volume you want it, and then you add the fusion. And if it's hot start, you add when the program hits 85. Okay, so let's talk about the program because there's a couple things to notate about this program. So it's a standard PCR program. So now we're actually running the reaction in the thermocycler. Okay, this is standard. 98 initial denature for 2 minutes. I did the hot start, so I cooled it down to 85. I paused it, I opened it up, I loaded my fusion, closed it, and then restarted. Okay, now it's starting to loop. 98 is the denature, 10 seconds, that's standard. 55, I just put 55 for my annealing. That's fine, that's standard. 72, 4 minutes, and 30 seconds. Why is that? So this is our, what step is this? Right. Why is it, most PCR reactions that you'll look at are only like 30 seconds. Why is this one so long? It's not because of the amount of primers. Think about the template.
Oh, it's running all the way around the plasmid.
It's got to run all the way around the plasmid. How big is a standard plasmid? 5,000 base pairs. That's a lot. Plus whatever, if you're inserting something or if you have an inserted gene, your plasmid might be 8,000 base pairs. So you're running all the way around. So if you look at this extension step, it's an oddity in terms of PCR reactions. And the reason it's an oddity, the reason it's so long, is because you have to PCR all the way around the plasmid. You sleeping?
Try not to.
Okay. Now, the other thing that's an oddity. So most PCR reactions, how many times do you go through this loop? 35 to 40 is what you usually see. Now, I'm telling you here, you cycle 25 times. Why? No, we can usually load enough DNTPs. I don't think we'd run out. There's a different reason. That could be happening. That's not a bad hypothesis. The real so that might be one of the reasons why you only do 25 times. But the real reason is that so what's the error rate? What's the error rate of fusion? Good, very good. 1 in 10,000. If we are doing SLIM of 7,000, let's make it easy on ourselves. Let's say we're doing SLIM on a 10,000 base pair plasmid. You're going to get 1 mutation every cycle. Okay? And every cycle you're amplifying exponentially the amount of DNA you have. So if every cycle you get 1 mutation in each molecule, that means by the end you have 25 mutations in all your products. Okay? Now if you run this 35 to 40 cycles, how many mutations you have? 35 to 40 mutations in every plasmid. Okay? Now, it's not such a big deal if those mutations are not in your gene or if they are in the backbone somewhere. But if they hit your AMP cassette, what's that going to do?
It won't survive.
It won't survive. You won't be able to select for that plasmid. If they hit your gene of interest, you may get the insert, but now you've also put a mutation that you did not want to have. Okay? So the reason it's 25 cycles is to kind of minimize the amount of mutational background that you are injecting into your plasmid. Okay? So that's one of the downsides of the SLIM method, is that it's a PCR-based method. All PCR-based methods are going to induce mutations. So here's the catch also, is that, is it going to work better on small or large plasmids? Yeah, it's going to work much, much better on small plasmids because you could PCR the whole plasmid without getting a single error if it's 5,000 base pairs, okay? Then you're only going to get an error every 2 times you PCR it. So it works much, much better on small plasmids. And this is a theme in molecular biology that I've been telling you over and over and over. You need to consider the size of things because if it's a 10,000 base pair plasmid, SLIM will never work. It just won't work. So it only works on small plasmids, and it usually works. So I've done it on things, I've seen it done up to 8,000. I've never seen it done on something higher than 8,000. And oftentimes, here's a trick you can do. Let's say you did it. Let's say you did it on an 8,000 base pair plasmid. You got it to work. It worked. You sequenced your gene, and it had your insertion. Okay? You would still worry that your backbone was mutated in some way that you did not want. So what you could do is you could subclone that mutated product. You could just cut it out and clone it into a new perfect backbone. Does that make sense? You could just subclone it. So you could do the mutation, pop out the mutation, and clone it into a backbone that you know is perfect. So that's how you can get around that, that you're mutating the backbone. But this is a consideration you need to be aware of. Okay, let's talk about this step right here. Because here we're going to start, now you're going to start to see why I've been teaching you guys about these enzymes. Okay, so what's DPN1? So there's a step where we add DPN1. Okay, so if you read the paper says the biggest problem for efficiency in SLIM is basically recovering the backbone. What does that mean?
Like your original backbone.
Yeah, you're basically selecting something that was not changed, okay? So imagine you're setting up a PCR reaction and what you're putting in there is a template backbone. It's a perfectly fine plasmid. And then you later transform this mixture, this PCR mixture, into E. coli. Is E. coli going to prefer the mutated plasmid or the one that's perfectly normal? The one that's perfectly normal. So one of the big problems is you're going to recover your backbone. And so you need a way to select for your mutant that you make. How do you select for it? You need a way to select for your mutant. How do you get rid of DNA that's made by bacteria? You add DPN1. So bacteria, when they replicate DNA, they methylate it. So your backbone comes from a mini prep. You grew that plasmid in bacteria, and then you purified that plasmid, and all that plasmid was methylated. And you put that into your PCR reaction. Okay, and then you're amplifying PCR around that methylated backbone. Okay, so question. Does the amplicon products that you're synthesizing around the plasmid, are they methylated? No. So DPN1 is a means that you can destroy the template that came from the bacteria, but not destroy the thing that you want. Okay, so it's a way to select for the plasmid that you want. So you'll see this commonly, DPN1, you'll see this pop up over and over and over and over again in molecular biology methods. And the reason is because DPN1 is the only way we have to get rid of a source template that we were generating from E. coli and select for something new that we've made. That's it. Let's end there, and then we'll start the lab. Thanks for catching that error. Does that make sense? Oh, wait. Actually, I just realized I needed to finish on Gibson cloning. I have 2 minutes. Okay, so Gibson cloning and overlap extension. So both these methods are ways to PCR something that produces sticky ends, okay, that can fuse with different products. So overlap extension is when you have X and you want to fuse it to Y, okay, and what you do is you make your primers encode a part that matches each, okay? Then what happens is you mix these things together, X plus Y, in a new reaction and do a new PCR, and then all of a sudden you have strange annealing patterns where you get X annealing to Y. Then your fusion polymerase will build a fused construct. Does that make sense? Do you see the logic behind that? So this is called either sewing or overlap extension. So you're designing primers that have overlaps, and the overlaps produce annealing, which causes things to get fused together in the PCR reaction. So this is very, very similar to SLIM. You're producing these overlaps. Gibson assembly is the same thing, except that the enzymes are different. So Gibson assembly, if you want to assemble, if you want to assemble X to Y, you still build the overlap and you build this into your primer. So this matches here. And then you add exonuclease, which comes on the end and degrades that and degrades that. Now all of a sudden this finds that and they link together. So it's a way to fuse PCR products by using an exonuclease. An exonuclease is something that comes in on the end and degrades DNA. Okay, so that's very, very brief. Watch the movie on watch the movie that I assigned. Okay, let's go to the lab.